You were built for this. Not the cosmos, not the void. This. The 9.8 meters per second squared pulling your skeleton into the ground right now. The atmospheric column pressing 101,325 pascals against every square centimeter of your skin. The magnetic field threading through the planet's iron core, wrapping around you like a cage, invisible, essential, non-negotiable. You did not evolve toward the stars, you evolved against them. Every system in your body is a negotiation with this specific planet. The density of your bones, the pressure of your blood, the architecture of your cardiovascular system, optimized over 300 million years of vertebrate iteration to move fluid upward against a fixed, reliable gravitational constant. Remove that constant, and the negotiation collapses, the contract breaks, the hardware begins to fail. NASA calls it Spaceflight-Associated Neuro-Ocular Syndrome. Astronauts call it blurry vision and pressure headaches. The correct term is systemic incompatibility. The human body in space is not adapting, it is degrading, methodically, on a schedule. We have been told a story. The story goes like this. Humanity is a species of explorers. The stars are our destiny. The cosmos is our inheritance. Columbus sailed west. Armstrong walked on a gray rock. Therefore, the galaxy is ours. This is a mythology. It is a product of marketing, of geopolitics, of the peculiar human need to believe the story ends well. The data does not support it. Earth is not a cradle. The cradle metaphor is sentimental, and sentiment is a form of data corruption. Earth is a highly specific biological habitat, a narrow band of temperature, pressure, radiation shielding, and gravitational force, a band so precise, so improbably calibrated that life outside it does not persist. It degrades. It fails. We are not trapped here by lack of imagination. We are trapped here by molecular biology, by the double helix, by the cell membrane, by the photoreceptor and the glomerulus and the trabecular bone. The prison is not around us. The prison is us. The question is not when we reach the stars. The question is whether the thing that arrives, if anything arrives, can still be called human. The answer, examined honestly, is no. The brain floats. This is not a metaphor. The human brain is suspended in cerebrospinal fluid inside the skull, a hydraulic system, pressure-balanced, gravity-dependent. On Earth, roughly 150 milliliters of CSF circulate continuously, flowing downward, draining, recycling, maintaining intracranial pressure within a precise operating range. Remove gravity, the fluid does not drain. It redistributes, it pools, it shifts cephalad, headward, flooding the cranial vault with pressure the system was never designed to accommodate. This is not a theory, this is observed data from the International Space Station, accumulated across decades of human subjects who volunteered to be the experiment. Here is what happens. Within 24 to 48 hours of microgravity exposure, astronauts report what they describe as a head cold that never clears. This is the cephalad fluid shift, blood and CSF migrating upward, the sinuses filling, the face puffing, the legs thinning, the body redistributing its liquid mass as though the floor has disappeared, because it has. Within weeks, intracranial pressure begins to rise. The optic nerve, a direct extension of the brain, sheathed in CSF, begins to compress. The eyeball flattens. Visual acuity degrades. Astronauts on 6-month missions frequently return with permanently altered vision. On longer missions, the degradation accelerates. NASA's VIIP study, Visual Impairment Intracranial Pressure, identified this as one of the most significant risks for deep space missions. Not asteroids, not equipment failure. The brain pressing against the eye from inside the skull. But structural damage is not the complete picture. Cognitive function declines in space. This is measured. Memory consolidation weakens. Processing speed drops. Spatial reasoning, the exact cognitive function you would need to navigate an alien environment, degrades under chronic stress, radiation exposure, and sleep disruption caused by the absence of a natural circadian cycle. Without the sun rising and setting on a 24-hour schedule, the human body's internal clock drifts, sleep architecture fragments. The glymphatic system, the brain's waste clearance process, active during deep sleep, operates at reduced efficiency. The brain, in space, is slowly filling itself with its own metabolic waste. Researchers studying astronaut cognition post-mission have documented measurable changes in white matter, the connective tissue of the brain, lesions, microstructural alterations, changes that, in any clinical context on Earth, would be characterized as early neurological damage. We do not call it that in press releases. We call it adaptation. We call it the challenges of spaceflight. The clinical term is hardware degradation. The functional term is your brain is being destroyed at a rate proportional to time spent away from the environment it was built for. A Mars mission takes 7 to 9 months one way, minimum 18 months on the surface, 7 to 9 months return. Add pre-mission preparation and post-mission recovery. That is approximately 3 years of continuous neural degradation. 3 years of rising intracranial pressure, disrupted sleep architecture, cognitive decline, and vision loss. And that is the optimistic timeline. The Earth has a magnetosphere. You have never thought about this because you have never had to. You live inside it. It is invisible, omnipresent, and the only reason your DNA has not been shredded. The magnetosphere deflects the solar wind, a continuous stream of charged particles from the sun moving at 400 to 800 kilometers per second. Without it, the solar wind would strip the atmosphere, would irradiate the surface, would make complex life statistically impossible. It is, functionally, the lid on the container that keeps you alive. Leave low Earth orbit and the lid comes off. Beyond the magnetosphere, 2 primary radiation sources become the dominant threat to biological continuity. First, solar particle events. Sudden, massive bursts of high-energy protons ejected during solar flares or coronal mass ejections. Unpredictable, intense. A significant SPE in open space without adequate shielding delivers a lethal radiation dose within hours, not over a lifetime, within hours. Second, galactic cosmic rays. This is the quiet killer. GCRs are high-energy particles, primarily protons and atomic nuclei, originating outside the solar system, accelerated by supernovae and other high-energy astrophysical events. They travel at near-relativistic velocities. They penetrate conventional shielding. They pass through metal. They pass through water. They pass through flesh. And when they do not simply ionize. They cause clustered DNA damage, multiple strand breaks in close proximity that the cell's repair mechanisms cannot process efficiently. The result is chromosomal aberrations, misrepaired breaks, and mutations. These mutations accumulate. They do not reverse. They propagate. Cancer in this framework is not a risk, it is a mathematical certainty scaled to exposure duration. NASA's own risk models, the REID models, risk of exposure-induced death, estimate that a round-trip Mars mission would expose crew members to radiation doses exceeding current career limits by a factor of 2 to 3. These are conservative estimates, They assume average solar activity. They assume no major SPE event in transit. They assume luck. Luck is not a life support system. The physics compounds the problem. GCR shielding requires mass, significant mass. To reduce GCR exposure to acceptable levels on a deep space vessel would require shielding so heavy that current propulsion technology cannot lift it. The solution to radiation requires a ship we cannot build. The ship we can build irradiates its crew. This is not an engineering problem awaiting a solution. This is a physics constraint operating at the boundary of material science. The human body, it must be noted, is an extraordinarily poor radiation shield. It is approximately 60% water and carbon compounds. These materials, under heavy ion bombardment, generate secondary radiation, a cascade effect where the initial particle impact produces additional harmful particles inside the tissue. In some scenarios, thin shielding is functionally worse than no shielding. The ship itself becomes a radiation multiplier. Consider what this means for the narrative of space colonization. Every mission beyond low Earth orbit is, in cellular terms, an ongoing act of molecular self-destruction. Every day in transit is a day of DNA corruption accumulating in the genome. By the time the crew reaches Mars, they have already been compromised, not metaphorically, biologically, at the level of the chromosome. The data does not negotiate with the mythology. Bone is not static. This is the first thing to understand. Bone is a living tissue in constant dialogue with mechanical stress. Osteoblasts build it. Osteoclasts break it down. The balance between these processes is calibrated by load-bearing, by gravity, by movement, by the mechanical pressure of a body operating in a gravitational field. Remove the field, the dialogue ends. Osteoclasts win. Bone is resorbed faster than it is built. Density drops, structure weakens. The skeleton without gravity is dissolving. Astronauts on the International Space Station lose approximately 1 to 2% of bone density per month in weight-bearing regions: the hip, the spine, the femur. 6 months in microgravity produces bone loss equivalent to decades of age-related deterioration on Earth. The spine elongates as vertebral discs decompress. Back pain becomes near universal. Herniation risk escalates. Exercise protocols mitigate this. They do not stop it. Current ISS countermeasures, resistance exercise 2 hours daily, slow the rate of bone loss. Astronauts still return with structural deficits. After 6 months, bone density in some individuals does not fully recover after a year of rehabilitation. After 12 months, the recovery curve extends further still. A Mars mission is not 6 months. It is 3 years. Extrapolate the bone loss curve over 3 years of microgravity and partial gravity. A Mars arrival crew may have skeletal structures too compromised to function effectively in even Martian gravity, approximately 38% of Earth's. The mission has not begun, and the crew is already partially disabled. The musculature follows the same trajectory. Without load-bearing, fast-twitch muscle fibers atrophy preferentially. The cardiovascular system deconditioning. The heart, a muscle, also weakens. Stroke volume decreases. Orthostatic intolerance develops, meaning that upon return to a gravitational environment, blood pressure regulation fails, and standing up becomes a medical event. Astronauts returning from long missions routinely cannot walk. They are carried off the spacecraft. They require weeks of rehabilitation to perform basic locomotion. This is the human body after 6 months in space, the optimized specimen selected from millions of candidates, exercised for 2 hours every day, monitored by the most advanced medical technology on Earth. After 3 years, After 5? After the proposed timeline of a permanent Mars settlement? The outcome is not adaptation. It is progressive systemic failure. The body is a machine calibrated for one specific operating environment. Operate it outside that environment long enough, and the machine breaks down. This is not complexity. This is not nuance. This is engineering.
The species problem. Everything discussed so far applies to individual bodies, to n equals 1, to the astronaut, the crew member, the test subject. But the question of space colonization is not the question of individual survival. It is the question of species persistence, of reproduction, of whether the second generation is even theoretically possible. The data here is not incomplete. The data here is almost entirely absent because we have not yet conducted the experiment. And the experiment we have conducted on model organisms is not encouraging. Mice embryos fertilized in microgravity show developmental abnormalities. Cell division without gravity produces irregular orientation of mitotic spindles, the cellular machinery that separates chromosomes during division. Get the chromosome separation wrong, and the result is aneuploidy, cells with the wrong number of chromosomes. Aneuploidy is a fundamental mechanism of cancer. It is also incompatible with normal development. Nematode reproduction in space shows disrupted germline function. Plant reproduction under high radiation conditions produces increased mutation rates in pollen and seed. These are proxy organisms. They are not humans, but they share the same molecular biology, the same DNA double helix, the same mitotic machinery. Human reproductive biology has never been studied in space because ethical constraints prevent it. We do not irradiate human embryos. We do not implant them in uteri aboard spacecraft. We do not conduct the experiment. But we know the component processes are compromised. Male astronauts show altered hormonal profiles. Testosterone levels fluctuate under chronic stress and sleep disruption. Sperm DNA fragmentation increases under radiation exposure. Female reproductive cycles are disrupted by zero-G and radiation. And even if fertilization were achieved, even if the embryo divided correctly, fetal development requires gravity. The vestibular system, the cardiovascular architecture, the bone and muscle formation of a developing human, all of these processes depend on gravitational orientation. A human fetus developed in microgravity would not be a normal human fetus. What it would be is uncharted territory. But the developmental biology literature does not give grounds for optimism. On Mars, gravity is 38% of Earth standard. This is an improvement over 0. It is not sufficient. No one knows what a human child gestated on 0.38 G would look like, what its bone density would be, what its neurological architecture would produce. The first generation born on Mars would be, in biological terms, a hypothesis. And that hypothesis cannot be tested without creating the child and observing the outcome. There is no ethical path to this experiment. There is only accident or authoritarianism. The species cannot reproduce off-world, not reliably, not safely, not currently. This is not a small problem. Colonization without reproduction is not colonization. It is a forward operating base that requires continuous resupply from Earth. It is not independence. It is not civilization. It is a slow, expensive way to die somewhere else. So we arrive at the solution, the only solution the data permits. If the human body cannot survive space, and the data suggests it cannot, not over generational timescales, then the human body must be replaced. Not repaired, not supplemented, replaced. This is where the conversation becomes uncomfortable, which is precisely where it must go.
CRISPR and its successors have demonstrated targeted genomic editing with increasing precision, base editing, prime editing. The toolkit for rewriting the human genome at specific loci is advancing along an exponential curve. The question is not whether we can modify the genome, we can. The question is what modifications space would require and whether the organism that results still qualifies as Homo sapiens. Consider the requirements. Radiation resistance. GCR bombardment requires cells with enhanced DNA repair mechanisms, upregulated photolyase activity, improved non-homologous end joining, reduced apoptotic response to strand breaks. Organisms like Deinococcus radiodurans, a bacterium capable of surviving 1.5 million rads without losing viability, possess redundant DNA repair pathways and condensed chromosome organization. Engineering these pathways into human cells is theoretically achievable. What it produces is not a human cell in any classical sense. Bone loss requires either a modified skeletal biology, altered osteoclast suppression, modified load-sensing mechanisms, or abandonment of calcium phosphate bone structure entirely. Radiation hardening, metabolic redesign, vestibular recalibration for non-Earth gravity. Each modification cascades into the next. The genome is not a list of independent settings, it is a network. Change one parameter, and the network redistributes. Unintended consequences propagate. To build an organism that survives interstellar space, you are not patching the human genome. You are rewriting it. You are producing a derived form, a post-human substrate with human ancestry and increasingly non-human biology. The philosophers will debate whether this entity has continuity of identity with the original. The biologists will note that at some threshold of genomic divergence, you have produced a new species. The ethicists will observe that engineering a being into existence whose entire biology is optimized for an environment hostile to their ancestors raises questions that have no clean answers. But the alternative is no humans in space, full stop. There is no moderate position here. The data does not offer one. Either we accept the terminal incompatibility of the current human form with space and remain on Earth, or we accept that the entity we send to the stars is not us. It is our replacement, our successor, something we built to go where we cannot. We would be manufacturing our own obsolescence, creating the organism that inherits the cosmos while we remain behind or disappear entirely. This is not science fiction. The genomic tools are real. The timeline is uncertain, but the direction is not. The question is whether the civilization that creates the post-human space colonist considers that a success, whether the entity that arrives at Proxima Centauri in 300 years, modified beyond biological recognition, looks back at Homo sapiens with something resembling continuity, or looks back at us the way we look back at Australopithecus. Interesting, foundational, gone. The stars are not waiting for us. They do not register our ambition. The cosmos is not hostile to human life. That would imply it is aware of us. It is not. It is simply structured in a way that makes human survival statistically irrelevant. We are a biological accident on a warm, wet rock at the edge of an unremarkable galaxy. The universe did not produce us intentionally. It will not preserve us sentimentally. The data, examined without mythology, produces a single conclusion. The human body, this body, the one you occupy right now, has a maximum operational range of approximately 400 kilometers above sea level, which is the orbital altitude of the International Space Station, maintained with constant resupply, constant monitoring, constant medical intervention, and a return window measured in months. Beyond that threshold, systematic, irreversible biological degradation begins and does not stop. We have spent 60 years in space. We have sent 600 human beings beyond the atmosphere. The total cumulative time spent by humans in space is approximately 150 person-years. This is the full extent of the experiment, and the experiment has produced bone loss, neurological damage, vision impairment, DNA mutation, immune dysfunction, cardiovascular deconditioning, and psychological deterioration. These are not anomalies, they are reproducible results. The mythology of space colonization depends on a future technology that does not exist, solving problems that may not have solutions within the constraints of human biology, within a timeline that becomes less plausible the more carefully you read the literature. It depends on radiation shielding that physics has not provided, on propulsion that chemistry cannot currently achieve, on reproductive biology that has not been demonstrated, on a species that has shown repeatedly that it cannot survive outside its narrow habitat band. The romanticization of space, the Sagan-esque pale blue dot poetry, the Musk brand civilizational urgency, the cultural narrative that the cosmos is our birthright, is a story we tell ourselves because the alternative is difficult to accept. That we are local, that we are fragile, that the universe is not structured around our persistence. This is the terminal quarantine, not imposed from outside, encoded from within. The dirt is not our limitation. The dirt is our reality. The gravity pulling at you right now is not a constraint to escape. It is the operating condition of the machinery. Turn it off, and the machinery fails. Perhaps the post-human will go. Perhaps some engineered derivative of us, something we made, something we are not, will one day walk on a world orbiting another star. Perhaps it will look up at a different sky and feel nothing in particular because we engineered the anxiety out of it, along with the calcium-poor bones and the radiation-vulnerable DNA. That organism will not be us. Its success will not be our success. It will be our replacement completing a project we began and could not finish. We looked at the stars and called them a destination. The stars did not look back. They never do.